listeners. Welcome to episode 89 of Cell and Gene, the podcast. I'm Erin Harris. Thanks for your time. In just a sec, you'll hear my conversation with Adaptimmune's SVP of late-stage development, Dennis Williams. Dennis and I get into the groundbreaking world of TCR T-cell therapies with a particular focus on the Tecelra trial. You'll hear us talk through the strategies that are being explored to improve TCR T-cell persistence and efficacy in the immunosuppressive solid tumor microenvironment. We even break down the key differences between TCR T-cell therapies and CAR T-cell therapies. I'm excited for you and what you'll take away from this episode for yourself and your colleagues and peers on what are the promising outcomes associated with TCR T-cell therapies. And so with that, here's my conversation with Adaptimmune's Dennis Williams. Enjoy. Hey, Dennis, welcome to Cell and Gene podcast. Thanks for being here.
My pleasure. Happy to be here.
Good. Okay, so tell us a little bit about Adaptimmune.
Adaptimmune is an integrated cell therapy company that is, our focus is to design, develop, and deliver therapies to transform the lives of people with cancer. Our main focus is in the solid tumor space, and we predominantly in our late stage pipeline use TCR therapies to target various antigens that are expressed in a variety of cancer types. We have a fully integrated platform, so we have early pipeline research, we have early phase development in translational sciences, we have a late stage development space which I look after, and we have a commercial organization now that we've become a commercial organization, and we also have a complete manufacturing platform. So we can make lentiviral vector and our manufacturing site in the UK, as well as making a T-cell product here at our Philadelphia Navy Yard facility.
Excellent, excellent. I know about probably, I bet you it's 6 years ago now. I'm in the greater Philadelphia area, and I toured the Adaptimmune facility. And gorgeous facility, really nice location, certainly. And I can't believe it's been 6 years. I feel like I need to get back there soon. But anyway, that's a conversation for later. So I want to talk a little bit about the FDA and its granting of Adaptimmune's therapy, formerly known as Efficacy, and it's going to be sold as Tecelra. Now, it received accelerated approval for use in some people with metastatic synovial sarcoma who previously received chemotherapy. So I'd love to hear from you. What were the most important learnings from the phase 2 trial that led to Tecelra's approval? And then maybe what were some of the key challenges as well that you faced during the clinical development of it? And perhaps even, in particular, targeting solid tumors and how you overcame those challenges.
Yeah, certainly. I think we have to start a little bit about what the disease is like, right? So in this case, metastatic synovial sarcoma is a very rare cancer. It typically presents as a soft tissue mass in one of the limbs, maybe your legs, maybe your arms, often presents behind the knee. And unfortunately, it's often not diagnosed because people think it's an orthopedic injury or something of that nature. And it has a very high propensity to spread. Typically, the number one site of metastases is in the lung. And unfortunately, this is what takes people's lives. Given that it's a rare cancer, there has not been a lot of research for some of these treatments. And soft tissue sarcomas are a very heterogeneous group of diseases. And in fact, the standard of care therapy for that disease that we still use, right, it came out in the 70s, right? I mean, so there hasn't been a lot of really notable advances. And the reason I give this backdrop is there really was a pent-up demand for new therapies. So when we started the Spearhead-1 trial, we had a track history with people that treat this disease because we were developing another product previously called Tecelra, which I'll talk about shortly. But and that program had shown really impressive activity in soft tissue sarcoma. So when we started the Spearhead-1 trial, there was a lot of enthusiasm for a novel treatment. And, despite the rarity of that cancer, the enrollment in that trial was actually very good, right? Because of the uniqueness of the disease, there is a concentration of the patients at these centers of excellence, these sarcoma centers of excellence, these tertiary referral centers. And a lot of those centers also had experience working in the cell and gene therapy space. So it was a good opportunity to leverage sites that knew how to manage, the incoming, cell material. They know how to manage toxicities. They know how to deal with, the different nuances of cell therapy and autologous cell therapy trial. And like I said, there was such a demand to try to have novel therapies in this space. I have to say, I would be remiss if I didn't talk about one of the challenges when we started that trial. That trial started in earnest, really at very late 2019, early 2020. And of course, what happened in 2020, we had the COVID pandemic, which really made some new challenges because a lot of centers were not having patients come in for research-related protocols. There were other challenges as well, as you may be familiar, that cytokine release syndrome, which is a common toxicity for both CARs and TCR therapies, is treated with a drug called tocilizumab. And tocilizumab is also used to treat cytokine release syndrome associated with COVID-19, right? So at times there was a shortage of tocilizumab that had to be managed. But we got through that. And I think one of the things that came out of that trial was a response rate that really hasn't been seen in this setting in probably ever, minus what we see with lete-cel. So afami-cel produced a really impressive response rate, and those responses were really durable, and it really created a lot of enthusiasm that when data was presented at various scientific congresses, whether it was at ASCO or at CTOS, there really was a tremendous amount of interest in really bringing this product to market.
Good. Okay. What about, how would you say your entire experience, even like you said, having gone through what you went through during COVID and beyond, how has the experience shaped Adaptimmune's approach to future clinical trial designs that you and your team might have, and the endpoint selection for TCR T-cell therapies?
Yeah, sure. I think one of the things that we think about in these trial designs is you have to start at the beginning of a patient selection, right? So this is a biomarker therapy, and all the patients require two biomarkers, right? They have to be tested to make sure they have the correct HLA allele. And they also have to be tested for the antigen in question, whether it's MAGE-A4 in the case of afami-cel or NY-ESO-1 in the case of lete-cel. So implementing an appropriate screening protocol or having a screening methodology in the pivotal trial is important, because that's the first step in identifying a patient that could be eligible for therapy. I think in our setting, in the late stage portfolio for the sarcoma related studies, we have had a lot of success in using a response rate as an endpoint. But there are other endpoints that we're certainly interested in, things that are more of the time to event setting, whether the progression-free survival or overall survival, where, there wasn't a comparator arm in this trial, but the overall survival that we see is very thought-provoking, because it does seem longer than what you would have expected in this population treated with conventional therapies. Another thing that I would say in the development space that is a key learning is, the manufacturing process is critical to these programs. And when you embark upon a registration-directed trial, there is always a when do you want to lock that process versus making, continuous process improvements, because every change that you make during that trial will undergo a certain amount of regulatory scrutiny. And this is always a push-pull because you want to make improvements, right, as technology improves. But at the same time, you don't want to do anything that may jeopardize the data you've already accrued on your existing manufacturing platform. So this is something that I think all developers that work in this space, this is something they wrestle with all the time.
Oh, yeah, for sure. Absolutely. In fact, we cover that exact topic quite a bit on Cell and Gene and even on Cell and Gene, the podcast, certainly. Um, and speaking of, I want to take a step back and have a kind of a more broad conversation. I do cover CAR T on Cell and Gene extensively, all aspects of it, and same with, you know, TCR cell therapies. What, from your perspective, what are the key differences between the two? So between TCR T cell therapies and then CAR T cell therapies.
Yeah, they do share a lot of similarities. Let me just start with the similarities, right? I mean, I think for the products that are out there today that are approved, there is a lot of commonality about how they're manufactured, right? They all use a viral vector, whether it's a retroviral vector or a lentiviral vector. All of them expand the cells that are genetically modified. All the products use some form of lymphodepletion chemotherapy as to sort of make space for the cells and make the environment more favorable for engraftment of the cells. All of them, in all those products, we look for the persistence of the cells and monitor persistence long term. I think one of the key differences from a mechanism of action is CAR Ts use a sort of an antigen binding domain and a clear stimulatory domain, and looking for surface antigens, right? So CD19 is like the perfect example, right? Where your B cells express CD19, you can modify these T cells to recognize that biomarker, and you could target a hematologic malignancy that is a B cell related malignancy. It is perfect in the sense of its mechanism of action, and the data has shown that both for the CD19 CARs and the BCMA CARs, they have really revolutionized the treatment of those hematologic malignancies. The challenge, I think, for solid tumors is there are very few solid tumors that express a surface antigen like CD19. And CARs are not really able to recognize an intracellular protein. So what the TCR technology does, because it basically uses the same machinery that all of our T cells right now are dealing with viruses or things of this nature, even oncogenic sort of transformations. That's how your T cells keep things in bay. You can genetically modify T cell to recognize a peptide that is intracellular, but through the MHC complex, it expresses that peptide, this antigen on the outside of the cell, and now it can be recognized. So this is true for oncogenic driver mutations that exist. It could be in viral mutations that like associated with HPV. It could be associated with the technology that we have in our late stage program, which is cancer testis antigens, which cancer testis antigens are antigens that are expressed in a variety of cancers, but they're not expressed in normal human tissue other than the testes and the placenta. So they're a good target that you could focus on a cancer without being overly concerned about on target off-tumor toxicity.
Perfect. Okay, thank you. I appreciate that. Speaking specifically about solid tumors, what strategies are being explored to improve TCR T-cell persistence and efficacy in the immunosuppressive solid tumor microenvironment, would you say?
There are a couple of different strategies that we're certainly exploring. For one, we start with a lot of translational science work that we try to look at what's happening in the tumor after infusion. In other words, if I think about do we see number one, acquired resistance, right? So someone that has a nice response and then loses it, what's the reason for that? For example, was it loss of persistence? Was it loss of antigen? Was there some other exhaustion parameter that took place? So that's one of the things that we look at, as well as whether or not there is reasons for primary resistance. So we see these therapies work extremely well in a variety of patients, but they don't work in every patient. Why is that, right? Is it because in some patients they may have had a different tumor microenvironment where they had more immunosuppressive cells, Treg cells, that may have limited the effectiveness of the therapy. So we do a lot of research in that regard. And then once we know that, then we can start designing one of two things. Either we can think about combination strategies where we can give another therapy in with the T cell that can mitigate that, or we can engineer the T cell in a second generation form to have something that would improve its survivability in the case of persistence. So we could engineer a T cell receptor product to, let's say, express IL-7 or IL-15 that may improve its survivability and have longer term persistence. There are a number of things that even in the tumor microenvironment where you could also modify the T cell for certain markers that in fact may make for a more favorable environment.
Good. Okay, thanks. Finally, before I let you go, I want to talk a little bit about off-target effects. We've dedicated entire Cell and Gene Lives, which are digital roundtable events that I admire. We've dedicated entire hours to this particular topic. And so I'm curious to find out from your perspective, how can we better predict and mitigate potential off-target effects and toxicities associated with TCR T-cell therapies?
Yeah, we have spent in our preclinical safety package, we do a lot of testing to try to identify whether or not the TCR is identifying, number one, an on-target off-tumor toxicity. In the case of the cancer testis antigens, that this is generally not likely. But then we also look at whether or not they can also identify a different peptide that is similar enough that it may recognize, and the T cells may start to react to that other peptide. So we do a lot of extensive preclinical testing to sort of identify any signals and whether or not those signals need to be investigated further. And we have had early pipeline research projects that we have terminated because we were uncomfortable that there may be sort of off-target toxicity. Even in the case of Tecelra, that it has a contraindication for HLA-0205. And the reason that is that preclinical testing demonstrated that you may say that its affinity under that HLA allele is so potent that the TCR may actually start to recognize peptides that were unintended, right? So this is definitely an area that we think a lot about. And previously we had a program in hepatocellular carcinoma that targeted alpha-fetoprotein. And alpha-fetoprotein, unlike a cancer testis antigen, this is an antigen that your normal liver can express. So it's not unique to malignant liver cells, although in that disease, the hepatocellular carcinoma expresses a lot of that, which made it a good target. And in that case, what we ended up doing with that program is designing a TCR that actually was less potent than we would have traditionally targeted. To see that we would have the right risk-benefit balance of targeting that cancer cell, but not causing undue toxicity to someone's healthy liver tissue. And we had good success with that endeavor. I would say in that program, the potency against the hepatocellular carcinoma, probably because of that endeavor, was not quite where we wanted it to be. So we actually discontinued that program. Ultimately, it's one of the things that we spend a lot of time in the clinical trials looking for any evidence of off-target toxicity. And unfortunately, despite the best efforts in preclinical research, some of this will need to be further evaluated in a clinical trial to see whether or not, in what setting you see it, its frequency, its severity, and what are the best strategies to mitigate.
Sure. Okay. All right. Well, listeners, that wraps up this episode of Cell and Gene, the podcast featuring Dennis Williams, Senior Vice President of Late Stage Development at Adaptimmune. Dennis, thank you so much for your time. This was wonderful. I appreciate your time and your insight.
Thank you. Happy to be here.
Good. All right, listeners, we'll talk to you soon.